Hello, in this video I'll be talking about the processes of muscle contraction and also the different types of contraction the muscle can perform. There are three main steps towards muscle contraction: nerve transmission, the neuromuscular junction, and the sliding filament theory. These all occur in order and result in movement of the muscle. So these three steps, are always occurring in day-to-day life, but they also occur in sporting environments. An example I'll be referring to throughout the video is a ball coming in towards goal and the goalkeeper moving to save it. The ball acts as a stimulus in the environment and the goalkeeper is the response. What I'll be explaining is the steps in between which allow for this response. Okay, so when the stimulus, the ball, comes in, it is picked up by a receptor, in this case the retina of the eye. The eye will then send a nerve impulse down a sensory neuron to the central nervous system, which in this case is the brain. The impulse basically tells the brain about the stimulus. The brain then decides what to do, as it sends impulses around the central nervous system via relay neurons to come to a conclusion about what to do. Once it has come to a conclusion, it will send another impulse down a motor neuron telling the muscle it needs to contract. When the impulse sent from the central nervous system reaches the muscle, it has to cross a synapse between the end of the motor neuron and the muscle fiber. When the transmission gets to the synapse, the motor neuron releases a chemical called acetylcholine, which is stored in vesicles in the motor neuron. The ACH will then diffuse across the synapse and bind to ACH receptors on the muscle fiber. If enough ACH gets across the synapse, the transmission can carry along the fiber's membrane, the sarcolemma, and then enter the sarcoplasm via T-tubules. When the muscle is at rest, it means there are no signals coming from the central nervous system telling the muscle to contract. This means that each sarcomere in the myofibrils take a particular shape. The actin, which is bound to the Z line of the sarcomere, stays in place and its binding sites are covered by troponin and tropomyosin, two proteins that act as bodyguards to the actin. The tropomyosin, which is bound to the M line, is connected to an ATP molecule, meaning it is an energy store waiting to be put into action so it can connect to the actin. It has an energy store because when one of the phosphates is broken off the ATP, creating ADP, energy is released. However, the myosin cannot connect to the actin as the binding sites are covered by troponin and tropomyosin. To initiate a contraction, the troponin and tropomyosin must be removed so that the myosin can connect to the actin. In order for this to happen, enough ACH must get into the sarcoplasm via T-tubules. If enough does get in, it opens up the sarcoplasmic reticulum within the sarcoplasm. These contain calcium, which will then flood into the sarcomeres in the myofibrils. These calcium ions bind with the troponin proteins, causing them to change shape. This change of shape causes them to move and therefore move the tropomyosin away from the actin binding sites, leaving it open to the myosin. So when the myosin is connected to the troponin and the actin binding sites are open, the myosin is free to connect to the actin. To do this, it must break a phosphate off the ATP, creating ADP and energy, allowing it to move towards the actin binding sites. When the myosin is binded to the actin, it pulls the actin towards the M line of the sarcomere, which is the middle line. This results in the two ends of the sarcomere, the Z lines, getting closer together. This happens all the way across the muscle and results in a contraction of the muscle. So once the myosin has performed the movement of actin, it needs to release so it can do it again. However, it can only release if it changes shape, and can only change shape if it has energy. Therefore, the myosin needs to bind with another ATP molecule and then break that molecule to gain energy and release from the actin. Once this has happened, the whole process starts again, and when the myosin binds to another ATP, it can attach to an actin again. There are four types of contractions that occur in muscles: concentric, eccentric, isometric, and isokinetic. I will be explaining how each of these are different. Concentric muscle contraction is when the ends of the muscle get close together. This is always in the upward phase in a movement. For example, in a bicep curl, when the forearm moves upwards, the ends of the bicep brachii are getting closer together. Or when someone stands up, the ends of the quadricep muscle group are also getting closer together. Eccentric muscle contraction is when the ends of the muscle get further away from each other. This is always on the downward phase of the movement. For example, again in a bicep curl, when the arm is lowered, the bicep brachii is performing eccentric contraction, as the ends are getting further away from each other and the muscle is lengthening. Isometric contraction is when the muscle is contracted but is staying still. For example, when holding a squat between the downward and upward phase, the muscle is keeping its position. It does this by a series of partial contractions and the muscle is still exerting force, but there is no movement occurring. Isokinetic contraction is where a machine is used to read the force a muscle will exert on an object. It works by setting a pace of contraction and will not change no matter how much force is exerted on it, as seen in the picture. A person must contract a chosen muscle part as hard as possible and a force reading is collected. It is then done for the opposite limb, so the other leg or the other arm. If the readings are very different, it means they have muscle imbalance, which can be bad for athletes because they need muscle balance to perform their sports correctly. Finally, I will be talking about motor units. Motor units are a nerve and any muscle fibers they control, as seen in the picture. Motor units can be slow oxidative or fast glycolytic. The structure of these two are different. Fast glycolytic motor units are quite large as they need more ACH to contract, as they are high power contractions and therefore need more neural branches. Slow oxidative is the opposite, as it needs less ACH to contract and therefore requires less neuron branches.